One of the places we are gaining a closer understanding of the extinct Neanderthal by bringing parts of him/her back to life is the Max Planck Institute for Evolutionary Anthropology, MPI-EVA. This institute is a world leader in genetic research into Neanderthals, our closest extinct relatives. In 2010, after years of DNA sequencing and research work, a team led by one of the Institute's directors, Svante Pääbo, published the genome of female Neanderthals who last walked the Earth around 40,000 years ago. All genomes decoded to date are from female specimens. This work won Pääbo the Nobel Prize in Physiology or Medicine in 2022. One of the most important discoveries made at the time was that Neanderthals had not really died out. In fact, all modern humans outside sub-Saharan Africa still carry genes of these early hominins. Hence, early modern humans must have interbred with them when they emerged from Africa to colonize the entire world. Since then, the MPI-EVA has consolidated its lead in early human research by proceeding not just to sequence other whole Neanderthal genomes, but also to analyze the DNA of the Denisovans. This archaic human species split off from the Neanderthal lineage at a very early stage and lived in Asia, in some cases alongside Neanderthals and modern humans up until around 50,000 years ago. It, too, left genetic traces in some modern human groups, namely the indigenous populations of the Philippines, Papua New Guinea, and Australia, which carry an average of 5% Denisovan DNA in their genomes. Crucial to the discovery of this hitherto unknown hominin was an approximately 70,000-year-old finger bone found at the Denisova Cave in the Russian Altai Mountains in southern Siberia, whose DNA was decoded at the MPI-EVA in 2010. No Denisovan skulls, let alone skeletons, have been identified to date. All we have is DNA from tiny new bone fragments that are periodically unearthed in the same cave in Siberia. Far more bones, a large number of well-preserved skulls, and occasionally whole sections of skeletons have been found in the case of Neanderthals. Their genome is, next to ours, the best researched of all prehistoric human forms. The fact that the Leipzig team has been able to grow archaic human brain cells, and even miniature organs, organoids, is the result of this comprehensive sequencing work and of a strong similarity to the blueprint of a modern human. The differences amount to a tiny fraction of a thousandth of an otherwise identical genome.